Right above the eyes, deep within the frontal lobes, sits a highly specialized density of tissue called the orbital frontal cortex. This region operates as the brain's primary calculation center for evaluating choices we didn't make, effectively quantifying the heavy cost of missed opportunities. For decades, standard psychological models categorized the resulting feeling, remorse, as a transient state. It was considered a temporary wash of biochemicals that would eventually clear out of the nervous system. But that temporary model fails to explain the physical data. Subjects experiencing intense, ongoing regret demonstrate long-term, observable alterations in how they evaluate future risks, long after the initial emotion fades. This discrepancy arises because persistent regret functions as a targeted biochemical catalyst. To understand this, we have to look at structural remodeling. This is a mechanism of neural plasticity, where networks physically alter their own anatomy in response to specific, repeated data inputs. When the orbitofrontal cortex calculates a severe deficit between a choice made and a better alternative, it begins to physically rebuild its own architecture to prevent it from happening again. We can trace exactly how this happens, tracking the life cycle of a fleeting thought as it hardens into permanent neurological hardware. The ultimate result is a physical footprint left behind by a missed opportunity, a structural alteration that overwrites baseline neurological functions and dictates all subsequent decision-making. In a healthy baseline state, the neural network topology of the orbitofrontal cortex processes normal, low-stakes choices smoothly across a wide, evenly distributed grid of pathways. The system algorithmically weighs variables, comparing potential outcomes to chart the path likely to yield the highest level of satisfaction. The processing sequence fractures the exact moment the brain registers a past decision as a mathematically inferior choice. This recognition initiates a biochemical error state within the network. This cross-section diagram reveals distinct, brightly colored markers appearing at a localized node, the biochemical signals of the error state. Rather than dispersing randomly, they travel along rigidly defined trajectories. Notice the contrast. Everyday signals are diffuse, while error signals burn through the exact same anatomy with high intensity. The heavier the cost of the missed opportunity, the denser the flood of signals. This localized flooding overwhelms the normal operating capacity of these specific pathways, priming the cellular structures for immediate physical alteration. Calculating a missed opportunity is therefore an active biological trigger. It initiates a cascade of physical tissue reactions that software-level processing alone cannot achieve. To see this alteration, we must transition past the network level and examine the microscopic gap between two individual neurons, the synaptic cleft. Persistent rumination is typically treated as a psychological concept, but mechanically it is simply the high-frequency, relentless repetition of those isolated error signals across this specific gap. This diagram shows high-frequency firing flooding the space between pre- and postsynaptic membranes with neurotransmitters. This bombardment places severe stress on the receiving receptors. In defense, the postsynaptic membrane thickens and whitens, synaptic hypertrophy. As the tissue expands, it rapidly multiplies its receptors, packing the membrane at double its normal density. By increasing receptor density, the biological and electrical properties of this specific connection are completely rewritten. This thickened structure permanently lowers the threshold required to fire the pathway. Even a very weak electrical signal can now easily bridge the gap. Because the threshold is now so low, this specific regret-associated pathway is biologically biased to fire faster and more easily than the unthickened neural pathways surrounding it. Normal, non-ruminative thought passes through the brain without leaving a structural trace, but repetitive error signals literally build their own infrastructure. Rumination forces a literal hardware update, bridging the gap between a transient psychological thought and permanent physical brain tissue alteration. When we pull back from that single altered synapse, we can observe the macroscopic impact on the newly reorganized architecture of the entire network. This diagram of the wider orbital frontal cortex shows thousands of hypertrophied synapses linking into a hardened anatomical structure, a permanent neural filter. Baseline pathways remain, but are functionally subordinate to this dominant grid. When new neutral stimuli enter the network, they must navigate this architecture. The filter acts as a gating mechanism. Because hardened pathways have the lowest threshold, new information is magnetically pulled into them, bypassing thinner, resistant routes. A flexible baseline network would have distributed these new inputs evenly to evaluate them objectively. Here, the restrictive routing forces new data down a predetermined track. The specific missed opportunities of the past have constructed the exact architectural pathways used to evaluate all future choices, removing neutrality from the brain's decision-making process. To grasp the total impact of this structural remodeling, we need to compare the final functional outputs of a baseline brain against one shaped by regret. This split-screen chart compares the two. On the left, the baseline orbital frontal cortex operates with one primary operational priority: optimizing data inputs to calculate the path of maximum satisfaction and reward. On the right, the remodeled network has an entirely different priority. Data is aggressively routed away from risk, and the output is exclusively pointed toward risk avoidance and survival. The neural filter explicitly suppresses satisfaction-seeking behaviors, simply because pursuing a high reward inherently requires taking a risk. Looking at the bar chart below, we see how this extreme risk aversion mechanism operates strictly as an evolutionary trade-off. The biological logic is highly conservative. The brain permanently trades away the possibility of future reward to guarantee absolute threat mitigation. It refuses to allow the possibility of another mathematically inferior choice. This is why deep-seated behavioral aversion cannot be easily reversed through therapy or conscious thought alone. A person cannot simply think their way out of a physical anatomical barrier. Reversing this process requires entirely new, equally intense biological catalysts to break down the hypertrophied synapses and rebuild the surrounding tissue. In the neurobiology of regret, a missed opportunity exists as a physical mandate, restricting human potential in the name of biological survival.